Okay, so good morning, all the participants, and good morning to Dr. Gaurav Dave. I am Dr. Elizabeth Robin on behalf of Navrajra University and GSBTM. We heartily invite you, sir, for the session. So now it gives me immense pleasure to introduce the expert for today. So he is Dr. Gaurav S. Dave, who is serving as an assistant research scientist at Pulses Research Station, Sardar Krishinagar Dantiwada Agriculture University, Sardar Krishinagar Banaskantha. He has an expertise in biochemistry. He has done his PhD from Sardar Patel University in Vallabh in 2008. He has a teaching experience. In PG, he has experience of 12 years. In UG, he has an experience of 4 years. And he has a research experience which starts from 2004. Now, to his credit, he has a lot of papers to his name. So he has research papers about 16, one review article, 3 book chapters, 1 book to his credit. He has 2 patent industrial design. He has 1 published patent and 2 patents have been submitted. He has also guided a number of students. So he has guided 2 PhD students and MSc students around 2, and he has a lot of government funded research projects which amount to 6 of the research projects, and he has a consultancy project about 2. So now I invite sir for the session and to give his inputs on the topic metabolism and its regulation. Over to you, sir.
Yes, thank you, madam. Uh, Today we'll discuss about the metabolism and its regulation. Uh, I am thankful to GSPTM and Navrajna University for giving me an opportunity to discuss about this topic of biochemistry, which is, we can say, the core subject, core topic of the biochemistry, metabolism. I hope I'll able to deliver and I will able to justify the topic to the students. So we'll start with the basic uh, terminologies about the metabolism. So it is all the chemical reaction done by the living organism. Virtually all chemical reactions are catalyzed by the enzyme. Now we talk about the metabolism. We generally define it as the sum and the collective results of the catabolism and anabolism. Here, the definition is limited to the biochemical aspects and the biochemistries. It is catalyzed by the enzyme because we focus only in the living organism. But there are many other reactions which are not driven by the enzyme, which are not under the control of the enzyme. So, in those cases, it is also a part of metabolism, but we called it as a uh, non-enzymatic reactions. And this non-enzymatic reactions in the life, in the body, in the living system leads to the various catastrophic effects, and it will lead to the disease and changes in the metabolism, which will lead to the end of the life sometimes. One of the the reaction is about the production of the amyloid products in the diabetic condition, where the high level of a glucose leads to the non-enzymatic glycosylation of the various protein. So it is also, we can say, the metabolism. But in general, in the living system, we called it as the the reactions, chemical reactions, which are controlled and catalyzed by the enzyme, and which is responsible for the uh, sustainability, which is responsible for the life in the organism. So, as it is the collection of the catabolic and anabolic reactions, the catabolism is the breakdown of substances. So simply, when we consume the food, it will be break down into the smaller units, let's say from starch to the glucose, from the protein to the amino acids, and the monomers and the simplest form. Then further, the glucose will be metabolized and will lead to the production of the carbon dioxide and the water, and finally the ATP will be produced. So catabolism is the breakdown, and mainly it occurs in the cytoplasm and in the mitochondria. Anabolism is another part of the metabolism. That is one of the segment, you can say, the synthesis of molecules used to build uh, molecules which are responsible for the cell growth and multiplication. Nutrients are also converted to their storage forms by anabolic processes. Many anabolic reactions occur in the cytoplasm. Cells usually employs different chemical reactions for the breakdown and synthesis of the same molecule. So, in the biochemistry, uh, if if somebody will ask that what is the biochemistry, generally uh, we have this trend to study the definition with the basics whenever we start to learn any new topic. So biochemistry is made up of the two words, bio and chemistry, and instead of we can say chemistry, we can say it could be a chemology, the study of chemical reactions. But since the history of the chemistry, the try suffix is given as it was related to the art, not to the science, and because of uh, the 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 historical definition, that those people who knew the art of chemicals, these people were known as a chemist or alchemist, and then the subject become the chemistry, the term given the chemistry instead of the chemology. And the chemicals which are in the biological system, uh, the study of chemical reactions. So through this the tradition, we call it as a biochemistry, and this metabolism is the part of, we can say, very uh, the, the core part of the biochemistry. So the next,
Excuse me, sir.
yes, hello.
Sir, your voice is audible, but it's like very low. So can you please come near the speaker and speak? I don't
Okay,
know if it's
okay.
only
Now,
with my device or what.
is it audible now? Now,
Yes, 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 sir. Yes, sir. Yes, sir.
Yes, yes, sir. Yes, sir.
Thank you, sir.
okay,
Yes, sir. Yes, sir.
okay. So now we'll start with the carbohydrate metabolism. Carbohydrate occupies the central role in the cell because most of the ATP energy is produced from the carbohydrate source, and its supporting role are played by fats and amino acids. And the star of carbohydrate metabolism is the glucose. So the whatever carbohydrate will consume in a food, it will be metabolized into the first simplest form glucose, and then glucose will be metabolized through the glycolytic pathway, and after that it will be transferred into the TCA cycle, the pyruvate and acetyl CoA, and then finally the uh, ATP produced in the cell. So in carbohydrate metabolism, we'll see how the glucose is broken down inside the cell to produce energy, and how it is stored, how is the level of glucose regulated in the blood, and the defects in the regulation of blood glucose leads to the diabetes. So that is the general metabolic disorder condition, and we all know about the diabetic condition. So where the level of glucose is not regulated in the body, that will lead to the this diabetic condition. So in the glucose catabolism, the parts that cells use to oxidize the glucose completely to the carbon dioxide, and it involves the 10 steps with the help of different enzymes, and these enzymes are regulated at different level, and finally the acetyl CoA will be produced, and it will be diverted to the TCA cycle. So pyruvate and acetyl CoA is the you can say the main molecules for the starting of the TCA cycle. So from pyruvate to the acetyl CoA, and then finally the other molecules will be produced in the mitochondria. So glycolysis is the sequence of the chemical reactions by which the glucose is cleaved to the two molecules of pyruvate, that is 3 carbon acid. So from 6 carbon sugar to the 3 carbon pyruvate, and also known as the glycolytic pathway or the Embeden-Meyerhof pathway, after the name of two German biochemists. So who proposed it in the latter part of the 19th century. So in the glycolytic pathway, which happens in the cytoplasm, is divided into the two phases mainly. One is the energy investment phase, where the ATPs are consumed in the formation of the intermediate molecules, and then the other part where energy payoff phase, that is the of the ATPs. So these are the phases where this happens. So in the first step, glucose is converted to the glucose 6-phosphate, and the enzyme involved is the hexokinase, and glucose 6-phosphate to the fructose 6-phosphate. That is second step, where the phosphoglucose isomerase enzyme is involved. Then fructose 6-phosphate to the fructose 1,6 bisphosphate. So that is third step, phosphofructokinase, and then for fructose 1,6 bisphosphate to the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That is catalyzed by the aldolase enzyme. So these steps are the energy investment phase, where the two molecules of ATPs are consumed in the first and third step. So whenever the ATP is involved in the reaction, it is like the we can say it resists the reversible reaction, where the flow of molecules from backward to the forward direction, that is reversible reaction, will be the that will be controlled mainly. So in the first step, glucose 6 carbon moiety converted into glucose 6-phosphate, 1 ATP molecule will be consumed, and that phosphate phosphorylation of the 6th carbon of glucose. Then in the second step, the glucose 6-phosphate is converted into the fructose 6-phosphate. Now this is reversible reaction, and any reaction which comes in the equilibrium, we say that the rate of forward and reverse reaction is almost equal. So that's why it comes in the equilibrium, and here it is driven by the ATP. So when flow of energy involved in the any reaction, the reversible reaction will be inhibited mainly. So this is isomeric reaction, where the isomerism occurs and glucose is converted into the fructose 6-phosphate. Then in the next step, the fructose 6-phosphate is converted into the fructose 1,6-bisphosphate. So when the phosphate attached to the non-successive carbon, then we call it as the bisphosphate, otherwise it will be diphosphate. So it is fructose 1,6-bisphosphate. First and the second, sixth carbon is phosphorylated, because sixth already phosphorylated in the first reaction, and the first carbon will be phosphorylated here in this reaction. So enzyme which catalyze this reaction is phosphofructokinase. Now next step, that is you can say the end of the first phase, energy investment phase, where fructose 1,6-bisphosphate is converted into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So two molecules are formed. If you see in the number of carbon, as you can see 1, 2, 3, 4, 5, 6. So the 1, 2 and 3 carbon is going to form the dihydroxyacetone phosphate, and the numbering will be reversed, and in the glyceraldehyde 3-phosphate, the 4, 5 and 6 number of carbon is converted into the glyceraldehyde 3-phosphate. So in the NET examination and other competitive examination, the questions kind of the which number of carbon from the fructose 1,6-bisphosphate will give rise to the which number of carbon in the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So either in the 2 marks or in the 4 marks, depending upon the type of questions. The questions many times the questions have been asked in the NET examination from these steps only. So it's all about the which number of carbon will go into the dihydroxyacetone phosphate, and the rearrangement of the carbon in the either in the dihydroxyacetone phosphate or in the glyceraldehyde 3-phosphate. And the questions related to the aldol condensation also. So almost every alternate year, every alternate examination, the questions about the aldol condensation and this related to these steps have been asked. In the dihydroxy, in the next step, the conversion of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate interconversion. So as you can see here, the glyceraldehyde 3-phosphate will go further in the reaction. So two molecules of the glyceraldehyde 3-phosphate will go in the next phase, that is about the energy payoff phase. So dihydroxyacetone phosphate will convert it into the glyceraldehyde 3-phosphate, and then the next step will occur. So the enzyme involved in this step is glyceraldehyde 3-phosphate dehydrogenase. So with the help of glyceraldehyde 3-phosphate dehydrogenase enzyme, the glyceraldehyde 3-phosphate will convert it into 1,3-bisphosphoglycerate, and one molecule of NAD plus will form a one molecule of NADH with the help of one phosphate. If you see here, two molecules of ATP is produced in the last step, and NADH molecule is going to produce in the sixth step. So anybody knows how many ATP molecule will be produced from the one molecule of NADH? Hello?
Sir, please repeat the question.
Yeah. How many ATP molecules will be produced from one molecule of NADH? How
3
many
ATP.
ATP molecules?
5. 3 ATP.
Sorry?
3 ATP,
Yeah,
sir.
3 ATP. Yes. Okay. Thank you. Then the 1,3-bis phosphoglycerate will react with the ADP, and this 3-phosphoglycerate molecule and 1 ATP molecule will be produced. ADP to ATP. And in the next step, 3-phosphoglycerate to 2-phosphoglycerate. So phosphoglycerate mutase will transfer the phosphate from the third carbon to the second carbon. And the enolase enzyme finally convert the 2-phosphoglycerate to the phosphoenol pyruvate. And finally, phosphoenol pyruvate converted into the pyruvate by the pyruvate kinase enzyme. Okay, is there any idea why the phosphate is transferred from third carbon to the second carbon? Any idea why it is so? If a cell will employ the reaction system where you can convert directly 3-phosphoglycerate to the phosphoenol pyruvate without transferring the phosphate from third to the second carbon. So we can save the two enzyme. If you will imagine the total protein amount of the enolase and the phosphoglycerate mutase will be involved throughout the life. If we consider the 60 years age of the average, 70 years now, 70 years of the average age of the human being, then you can imagine that how many crores of phosphoglycerate mutase enzyme will be produced in the cell just to convert the 3-phosphoglycerate to the 2-phosphoglycerate and then finally to the phosphoenol pyruvate. So we can save the protein also, we can save the time, and the final product will be also produced easily. So why this step is required? Okay, we'll keep this one aside. Glucose can be converted into directly pyruvate, right? Why this many 10 steps are required in the metabolism? Why the nature has selected this only 10 steps, not even 11 and not even 9, to convert the glucose into the pyruvate? Any idea? Any guess why this many steps are there just to convert one glucose into the 2 pyruvate molecule? So nature would have evolved the system which can break the glucose and form the 2 molecules of pyruvate easily. So we can save this many enzymes, protein. So cell has to do very less work to produce this many enzymes. Any idea? Anybody?
Sir, is it related to free energy changes?
You can say yes, free energy changes are also involved. Apart from that, okay, let me give you the hint to give the answer of this. Suppose you have a regular diet of five chapatis, right? And after taking your 5 chapatis, you feel satisfied. Your hunger is up to the 5 chapatis regularly. So you want to eat 5 chapatis. Now, do you eat directly? You take a 5 chapatis in your hand, put a sabji, dal, everything, and then you put it into the mouth, or you take step by step. Even in the single chapati, you take a piece of chapati, then you take a sabji and dal, and then you eat. So why these two processes are there? What happen if you will take directly? The 5 chapatis at a one stretch. Am I audible? Hello?
Yes, sir.
Yes, sir.
Yeah, okay. Okay, any idea? Okay, point is about the regulation. If we consume the our food in a small segments with the small steps, it will be easy to digest and easy to regulate. And if we consume the 5 chapatis in a single stretch, I don't think so that it will be good to digest, as well as the level of satiety will take time. When we consume the food, the digestion starts right from the our buccal cavity, from the mouth itself, and then it will be digested slowly, reach into the stomach, and then glucose will come into the blood, and then it will be sensed by the brain, and then brain gives signals that okay, the level of glucose is sufficient, and then you can stop. Sometimes what happens when you consume the sweet simultaneously with the regular diet, you will not able to take the regular diet as you are taking regularly, the quantity of the diet, because the sweet gives you the sufficient sugar, and then it will be sense, and your body will sense that okay, the level of sugar is sufficient, you don't require much energy now. So automatically the quantity of diet will be dropped down. Reason is, if you consume the 5 chapatis as a single stretch, level of satiety will not be sensed immediately in the blood. Same way here in the metabolic pathway, if you have a 10 steps, the different intermediates and different enzymes can be regulated with the minimum energy. Okay, let me put it in this way, in the reverse form. Suppose instead of a 10 steps, it's only a single step. So if anything happens, any metabolic disorder, any genetic changes or any mutation, entire glycolytic pathway, entire energy production will be stopped, and that will lead to the a very dangerous condition. But if there are 10 steps and there are different intermediates, it can be diverted into the different pathway, and total metabolic reactions will be regulated easily, as well as the chances of recovery will be more, chances of error will be less. Now here again counter questions can be asked that you have 10 different steps, so chances of errors are 10 times higher than the single step. But this chances of error will be even though it is higher, it will not completely uh, the we can say the catastrophic of the metabolic reaction. It will always give you the chance to correct it, and then to divert the different metabolism in the different part. So that is the main point. That's why we keep this 10 steps, and we transfer the different intermediates from one form to the other form. And same way here also, the pocket of the enzyme is designed in a way that the second position is fit, second position of phosphorylated molecule fit completely into the enzyme, and then it will be transferred into the ATP. So the pyruvate kinase enzyme has the specificity towards the second position, and that's why these different intermediates are formed, as well as the phosphoenol pyruvate and phosphoglycerate molecule. Two phosphoglycerate molecules has the different fate in the metabolic reaction. That's why. So the two molecules of ATPs are formed. Then phosphoenol pyruvate to the enol pyruvate, and then the enol pyruvate to the pyruvate. So this pyruvate molecule is the last molecule of the glycolytic pathway. So in the sixth step, two molecules of NADH are produced. Then in the next step, two molecules of ATPs are produced. Then in the ninth step, two water molecules are produced. And then in the 10th step, the two molecules of ATPs are produced. So this two molecules of ATP, again, it's the final, you can say, because the two ATPs are consumed in the first phase and four molecules are produced in the second phase, but the net ATP production will be 2. So 2 carbons of pyruvate are incorporated into the acetyl-CoA enzyme. And when an aerobic cell is operating with a good supply of oxygen, pyruvate molecule flows into the mitochondria. 2 of the carbons of each pyruvate ion end up as acetyl-CoA, and 1 molecule of NAD plus is reduced to NADH. So carbon dioxide is formed as waste product. So in the anaerobic condition, instead of this acetyl-CoA, lactate is produced. Which cell produce the lactate predominantly through the glycolytic pathway in the human body? Which cell?
Muscle, sir.
Muscle cell. So muscle cells do not contain mitochondria, is it so? Mitochondria are not present in the muscle? Or it is present, but it is not active, or
It is
any
present, but
defects are there?
it is
So
present
why
in
lactate
muscle
is
cells.
okay? Okay, muscle. Okay, fine. Lactate. Then any other cell? In muscle cell, when it is highly active in the exercise mode only, the lactate is produced. Otherwise, acetyl-CoA and the TCA cycle will be operated regularly. Means when muscles are resting, but only in the heavy exercise or muscular exercise, the lactate is produced. So there is also a reason behind this. Okay, which cell in the human body do not contain the mitochondria? Is there any cell in the body where mitochondria is not present?
RBC.
Yes,
RBC.
exactly. So RBC, yes, exactly. So RBC produce the lactate in the anaerobic condition, and in muscles, when we perform a heavy exercise, vigorous exercise, at that time the rate of we require a quick consumption of the ATP, and that's why the glycolytic pathway is operated, and the ATP will be produced immediately. And if acetyl-CoA will go into the the TCA cycle, then it will take time. So the rate of ATP which we require during the heavy exercise cannot be fulfilled. That's why. So multi-enzyme complex is responsible for the formation of the acetyl-CoA from pyruvate in the TCA cycle, which is operated in the mitochondria, in the inner mitochondrial membrane. So in the regulatory steps of the glycolysis, so there are irreversible steps, and these are regulated. And first one is the hexokinase or the glucokinase. So there are actually 6 types of hexokinases are there. Okay, what is the difference between the hexokinase and glucokinase or the fructokinase? Hexokinase is for the you can say general 6 carbon sugar monosaccharide, and glucokinase is specifically for the glucose. So what is the difference between these 2 type of enzyme instead of only the substrate? Glucokinase is absolute specific enzyme and hexokinase is the group specific enzyme. Okay, how you can identify what could be the parameter to characterize the enzyme? How you can characterize the enzyme? Do you know? Have you studied the enzymology? Okay, so the Km value, Michaelis-Menten constant, that is the integral characteristic of the enzyme. So glucokinase is a high Km for the substrate. That's why only when glucose level will go up, then only it can be activated and utilized. And hexokinase has a low Km value, so that's why it can easily react with the glucose and the other hexose and form the glucose 6-phosphate molecule. Then the phosphofructokinase 1 enzyme, where ATP is consumed and form the ADP in the step of a fructose 6-phosphate to the fructose 1,6-bisphosphate. Then, as we know that the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate is produced, and then the energy payoff phase operated. And finally, the pyruvate kinase enzyme, which is the regulatory step where the phosphoenol pyruvate to the pyruvate reaction will be regulated, the production of ATP in the last step. So glucose plus ATP, glucose 6-phosphate, that is the first step of hexokinase. So through the feedback inhibition by the glucose 6-phosphate enzyme, once the concentration of a glucose 6-phosphate will shoot up in the cell, it gives a negative feedback to the hexokinase enzyme, and then it will inhibit the reaction, and then finally the glycolytic pathway will be regulated. So as you can see in the graph, the in the y-axis the relative enzyme activity where hexokinase 1 and hexokinase 4, that is glucokinase, and its level, and the concentration of the glucose. So because of the low Km value of the hexokinase, even with the minimum amount of the glucose, it will remain activated, and the glucokinase enzyme require almost more than 8 millimolar concentration of the glucose. So that is the difference in the value of Km and the concentration of glucose required. In the feedback inhibition system, once the concentration of a product will rise above the level where it can react with the same enzyme, and it will bind to the other than active site, and then it will regulate the activity of enzyme. So in the hexokinase, the glucose 6-phosphate binds to the hexokinase enzyme on other than active site, allosteric site, and it brings the conformational changes in a way that the glucose cannot bind with the active site and the reaction will be inhibited. So this is how the hexokinase enzyme is regulated. Then phosphofructokinase 1, ATP is one of the negative regulator. AMP and ADP are the positive regulator. Same way citrate is the negative and fructose 2,6-bisphosphate is the positive regulator. So through the positive feedback inhibition. So glucose 6-phosphate reacts with the ATP and form fructose 1,6-bisphosphate. In this particular reaction, the allosteric effector molecules like ATP, citrate inhibits the reaction, and AMP, ADP, and fructose bisphosphate activates the phosphofructokinase enzyme. So this is how the activity of this enzyme is regulated. So it is obvious that in the presence of sufficient ATP, we do not require the metabolism of glucose, so glucose will be diverted to the glycogen synthesis. But when body requires the energy at that time, the level of AMP and ADP will be high. So once the level of AMP and ADP level will increase, that will activate the enzyme, and through this positive feedback mechanism, glucose 6-phosphate is converted into the fructose 1,6-bisphosphate. And once the level of ATP will shoot up, then it will give the negative feedback message signal. Same way for the citrate. So once the pyruvate will be formed, it will go into the TCA cycle, and then citrate, sufficient amount of citrate, that means the level of ATP and energy level is sufficient in the cell, so that citrate will give a signal to the enzyme for the inhibition. So if ATP level is low, activity of phosphofructokinase is high, and when ATP level is high, activity of phosphofructokinase 1 will decrease. So from this graph, we can say that low level of ATP for the high activity of PFK1 and high level of ATP is for the low level of the PFK1. So there are two binding sites, substrate and allosteric site, as it is an allosteric enzyme. So this regulator molecules always bind to the other than active site. So allosteric enzymes are those which is a two binding site. One is for the regulator and another one is for the substrate. So on the allosteric site, regulator molecule binds, and on the active site, substrate molecule binds, and then substrate is converted into the product. And when regulator binds, it regulate the, not inhibit, but it regulate, either it will increase the activity or it will decrease the activity. This is how the regulation occurs. And in the last step, that is control of the pyruvate kinase. So in the pyruvate kinase enzyme, phosphoenol pyruvate to the pyruvate, one ATP molecule is formed. So here the acetyl CoA is the negative inhibitor, and alanine molecule, because once the level of pyruvate increases, pyruvate is the precursor molecule for the alanine, and then it gives the feedback inhibition. So how the pyruvate kinase activity is regulated between the liver and other tissues. So in the liver, as you can see here in the picture, the pyruvate kinase enzyme, once it will phosphorylated, become inactive. So in the liver, the hormone glucagon is responsible for the conversion of ATP to the ADP and for the activity of pyruvate kinase enzyme. And then in the reversible step where the dephosphorylation occurs and pyruvate kinase molecule is formed. In the other tissue, the pyruvate kinase form the ADP to the ATP, and phosphoenol pyruvate is converted into the pyruvate. So this how the activity of pyruvate kinase is regulated between means it regulates the flow of phosphoenol pyruvate to the pyruvate and in the liver and other glycolytic tissues. So this is about the overall glucose metabolism. Glycogen to the glucose 6-phosphate, glucose to the glucose 6-phosphate, glucose 6-phosphate will go to the ribose 5-phosphate into the pentose phosphate pathway. Then glucose 6-phosphate will go further into the glycolysis and form pyruvate. Pyruvate to the glucose 6-phosphate by the gluconeogenesis. Pyruvate to the amino acid, lactate, acetyl-CoA and PCA cycle. So here you can say the glucose 6-phosphate is the central molecule which has a
Excuse me,
divergent.
sir.
Yes, yes. Hello.
Sir, your voice is too low.
Okay. Now is it
Yes,
audible?
yes, sir. Yes, sir,
Okay.
yes.
Yeah, I'm sorry. So glucose 6-phosphate is the here the central molecule where glycogen is formed. Then glucose to glucose 6-phosphate, glucose 6-phosphate to the ribose 5-phosphate, glucose 6-phosphate to the pyruvate amino acid, acetyl-CoA and lactate. And the acetyl-CoA will go into the citric acid cycle. So in the gluconeogenesis is the process whereby precursors such as lactate, pyruvate, glycerol and amino acids are converted to glucose. So in the gluconeogenesis process, the different molecules give rise to the glucose molecule when the body is under the deprived state of the ATP and glucose is not available. Fasting requires all the glucose to be synthesized from this non-carbohydrate precursor molecule. And most precursor must enter the Krebs cycle at some point to be converted to oxaloacetate. And oxaloacetate is the starting material for gluconeogenesis. So here in the TCA cycle, where the oxaloacetate molecule is formed, so from oxaloacetate molecule through the gluconeogenesis, the glucose molecule is formed. From pyruvate, also oxaloacetate, because it will go into the TCA cycle. From the amino acids to the lactate and pyruvate and then oxaloacetate, and into the TCA cycle also The different amino acid give rise to the different intermediate molecule. This all intermediate molecules will give rise to the oxaloacetate molecule, and then finally it will go into the gluconeogenesis and formation of a glucose. So in this step, pyruvate is converted to oxaloacetate before being changed to phosphoenol pyruvate. So in this reaction system, pyruvate carboxylase catalyzes the ATP-driven formation of oxaloacetate from pyruvate and carbon dioxide. So pyruvate to the oxaloacetate, and from oxaloacetate to the phosphoenol pyruvate. So in the first step, the pyruvate carboxylase enzyme, which convert the pyruvate into the oxaloacetate, and in the second step, this oxaloacetate is converted into the phosphoenol pyruvate. So now it is kind of the reverse reaction of the glycolytic pathway, where the phosphoenol pyruvate is formed. So PEP carboxykinase, that is phosphoenol pyruvate carboxykinase enzyme, that converts the oxaloacetate to PEP, and then that uses the GTP as a phosphorylating agent. Reason is, okay, which molecule gives the high free energy comparatively, GTP or ATP?
ATP, sir.
ATP. So ATP gives high free energy than the GTP. Is it so?
Sir, you are not audible at all.
Sir, your voice is not audible.
Sir, you are on mute.
Hello. Now is it audible?
Yes, sir.
Hello.
Yes, sir. Yes, sir.
Okay.
Yes, sir.
Give me a one minute. I'll be back. I think I got an issue in the network. Hello. Am I audible now? Hello.
Yes, sir.
Yes, sir.
Okay, okay, okay. So in the gluconeogenesis, which is not just a reverse of the glycolysis, but it produces the different intermediates, and then it will give rise to the glucose finally. So the
हेलो सर
several steps are different. Hello.
यस सर आप जीटीपी वाला रिपीट कर सकते हैं वहां से हमने सुना नहीं था
यहाँ से. Hello.
यस सर व्हाई वी यूज यहाँ पे जीटीपी क्यों है वो क्वेश्चन कर रहे थे ना आप
हाँ हाँ. So the reason is
वो आपने फ्री फ्री एनर्जी वाला पूछा ना वो
हाँ 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 free energy वाला. So the free energy changes require to convert the glucose into the phosphoenol pyruvate is higher, and that is not means fulfilled by the ATP. That's why GTP is required, because the changes in the level of free energy is the requirement is high, and that's why the GTP will be consumed, not the ATP. And GTP is having the high free energy change compared to the ATP. So there are several steps which are different, so that the control of one pathway does not inactivate the other pathway. And many steps are the same, and mainly the three steps which are different from the glycolysis is pyruvate to phosphoenol pyruvate, fructose 1,6 bisphosphate to the fructose 6 phosphate, and glucose 6 phosphate to the glucose. So in the phase 1, the ATP to the ADP and carboxyphosphate is formed in the biotinylated steps. So the enzyme will be biotinylated, and it will form the final active enzyme form. And in the phase 2, the pyruvate and the enzyme where the biotin molecule is attached will form the final reaction, and the oxaloacetate will be formed. Phosphoenolate and phosphoenolate to the oxaloacetate. So biotin is an essential nutrient, and there is hardly any deficiency for biotin because it is abundant in bacteria in the large intestine, also make it in the human body. However, the deficiency have been seen and are nearly always linked to the consumption of the raw eggs. Raw eggs contains the avidin, that protein that binds to the biotin with the dissociation constant of 10 raised to minus 15. That is one of the tight binding reaction, and that actually inhibits the absorption of the biotin from the intestine. And it is thought that avidin protects the egg from the bacterial invasion by binding biotin and killing the bacteria. So in a nature, avidin is to protect the eggs from the infection and bacterial infection. And if the raw eggs will be consumed, the avidin is responsible for the absorption, inhibition of the absorption of the biotin in the intestine. So phosphoenol pyruvate carboxynase enzyme, where the oxaloacetate converted into the phosphoenol pyruvate, and the GTP, the third phosphate, is going to be consumed here, and the reaction is there is a nucleophilic attack, and that nucleophilic attack will transfer the phosphate from the GTP to the oxaloacetate, and phosphoenol pyruvate will be formed. GDP and CO2 will be released. Acetyl-CoA regulates the pyruvate carboxylase activity. So increases in oxaloacetate concentration increase the activity of the Krebs cycle, and acetyl-CoA is the allosteric activator of the carboxylase enzyme. However, when ATP and NADH concentration are high and the Krebs cycle is inhibited, where oxaloacetate goes to the glucose. Now the transport between the mitochondria and the cytosol. Generation of the oxaloacetate occurs in the mitochondria only, but the gluconeogenesis occurs in the cytoplasm. So the phosphoenol pyruvate carboxykinase enzyme, which is distributed between both compartments in the human. So while in the mice, it is only found in the cytosol. So in rabbits, it is found in mitochondria. So that is the distribution of the enzyme in the different animals in the cell. Either phosphoenol pyruvate must be transported across the membrane or oxaloacetate has to be transported. Phosphoenol pyruvate transport system are seen in the mitochondria, but oxaloacetate cannot be transported directly in or out of the mitochondria. So in the gluconeogenesis reaction, how this transportation occurs across the membrane. As you can see here, the transportation between the mitochondria in cytosol and in the mitochondrial membrane. So there is a malate aspartate shuttle. This malate aspartate shuttle is operated to regulate the oxaloacetate flow across the membrane. So if we see first in the mitochondrial membrane, the reaction which happens, malate is converted into the oxaloacetate converted into the malate, and oxaloacetate is converted into the aspartate. So it gives rise to the two molecules, malate and aspartate, in the mitochondria. Now this in the route 1, the oxaloacetate converted into the aspartate by the aspartate aminotransferase enzyme with the byproduct of the alpha keto acid, and this aspartate crosses the membrane and reaches into the cytosol, where this aspartate further converted into the oxaloacetate by the aspartate aminotransferase enzyme. So alpha keto acid to the amino acid. So this oxaloacetate is now transported from mitochondria to the cytosol, and then the other steps will be followed and the glucose molecule will be formed. In the second route, oxaloacetate is converted into the malate by the malate dehydrogenase enzyme, and then malate will cross the membrane, will reach into the cytoplasm, and then further converted into the oxaloacetate by the malate dehydrogenase enzyme. So these two enzymes, aspartate aminotransferase and malate dehydrogenase, are present in the mitochondria as well as in the cytoplasm, and the oxaloacetate will be formed. This is malate aspartate shuttle for the transportation of the oxaloacetate, where phosphoenol pyruvate has the direct transportation without the conversion into the other intermediates, and phosphoenol pyruvate can cross the membrane and it will reach into the cytoplasm and it will go into the mitochondria also, and then it will give rise to the different intermediates into the gluconeogenesis, and the glucose will be formed. So in the hydrolytic reaction, which bypass the phosphofructokinase and hexokinase, the hydrolysis of fructose 1,6-phosphate and glucose 1,6-bisphosphate and glucose 6-phosphate are separate enzymes from the glycolysis. So glucose 6-phosphatase is only found in the liver and kidney, and the liver is primary organ for the gluconeogenesis. So the overall reaction will be glucose plus 2NAD plus 2ADP plus 2PI give rise to the 2-pyruvate, and from 2-pyruvate 2NADH 4 ATP, 2 GTP, and it forms the glucose. So overall, the 2 ATP, 2 GTP, and 4 H2O give rise to the 2 ADP, 2 GTP, and 4 PI. So here in the overall steps of the glycolytic pathway, where glucose to the glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 bisphosphate to finally the pyruvate. And in the last step, the phosphoenol pyruvate is converted to the pyruvate. Now in the gluconeogenesis, pyruvate is converted into the oxaloacetate, and from oxaloacetate to the phosphoenol pyruvate. Then now we'll go into the reverse direction and will reach into the the fructose 1,6 bisphosphate. So from fructose 1,6 bisphosphate to the fructose 6 phosphate by fructose bisphosphatase enzyme, and then from glucose 6 phosphate to the glucose by the glucose 6 phosphatase enzyme. So the red color step indicates the steps of the gluconeogenesis, where rest of the other steps are of glycolytic pathway. The values which are written in the into bracket in each step, that is the energy change required for the conversion of the one molecule to the other molecule, that is from the pyruvate to the phosphoenol pyruvate, and from fructose 1,6 bisphosphate to the fructose 6 phosphate, and from glucose 6 phosphate to the glucose. So in the first step it is minus 5.1, second step minus 8.6, and in the last step, that is third step, it is minus 22.6. Now, which are the regulators of a gluconeogenic enzyme activity? Here the formatting is little change. So the name of the activators and the inhibitors are uneven here in the column. So enzyme phosphofructokinase, that is ATP, citrate, AMP, and fructose 2,6 bisphosphate. Then fructose bisphosphatase AMP, fructose 2,6 bisphosphate in the phosphofructokinase, that is alanine. Then allosteric activity of fructose 1,6 bisphosphate, and it inactivates the enzyme. Then pyruvate carboxylase acetyl CoA. Then phosphofructokinase 2 inhibitor is citrate, activator is the AMP, fructose 6 phosphate phosphate, and enzyme phosphorylation inactivates the enzyme. And fructose 2,6 bisphosphatase 2, activator is fructose 6 phosphate, inhibitor is fructose 6 phosphate, activator is glycerol 3 phosphate, and the phosphorylation activates the enzyme. So if blood glucose level is low, it increases the glucagon secretion. Glucagon secretion increase the cyclic AMP production, that is secondary messenger. Primary messenger is the hormone, secondary messenger is cyclic AMP, another is the DAG and calcium. So here even inositol 3 phosphate. So the increase in the cyclic AMP concentration increase the enzyme phosphorylation, because it activates the phosphorylating enzyme tyrosine kinase. Activation of fructose bisphosphatase 2 and inactivation of the phosphofructokinase 2. So it will decrease the fructose 2,6 bisphosphate molecule. This will inhibit the activity of phosphofructokinase, and it activates the phosphate fructose bisphosphatase enzyme, and this how it will increase the gluconeogenesis process. So in the low blood glucose level, through the glucagon and through the signal transduction mechanism, finally the level of the rate of gluconeogenesis increases, that will give rise to the glucose molecule. So this is the hormonal, this picture is about the hormonal control of the fructose 1,6 bisphosphate and fructose 6-phosphate flow in the cell. The central molecule is fructose 2,6 bisphosphate. So fructose 6-phosphate converted into the fructose 1,6 bisphosphate by the enzyme phosphofructokinase 1. Then with the help of phosphofructokinase 2, it will give fructose 2,6 bisphosphate. Now fructose 2,6 bisphosphate is positive regulator for the phosphofructokinase 1. So phosphofructokinase 1 is for the synthesis of fructose 1,6 bisphosphate. So the flow of glycolysis will be operated. AMP is the positive regulator. ATP and citrate is the negative regulator. So when level of ATP and citrate is high, phosphofructokinase 1 activity will be inhibited. So the rate of glycolysis will be decreased. Now this fructose 1,6 bisphosphate to the fructose 6-phosphate. Here the AMP level, so low AMP level, that means the negative feedback inhibition. So conversion of fructose 1,6 bisphosphate to the fructose 6-phosphate will be inhibited by the enzyme fructose bisphosphatase. In the upper reaction, that is about the regulation of the activity by the cyclic AMP and protein kinase. So phosphofructokinase 2 enzyme is responsible for the formation of this fructose 6-phosphate and fructose 2,6 bisphosphate. Here in this reaction, protein kinase, cyclic AMP dependent protein kinase, phosphorylate the enzyme, and then it will inhibit the reaction. So fructose 6-phosphate inhibits the reaction, and phosphorylation of the enzyme is responsible for the inactivation. AMP is positive regulator, fructose 6-phosphate is positive regulator, and citrate is negative regulator, because this step requires the glycolytic pathway to be operated. And in the other side, where fructose 2,6 bisphosphatase enzyme is there, it will converted into the fructose 6-phosphate from the fructose 2,6 bisphosphate, and phosphorylation activates the enzyme, and fructose 6-phosphate is the negative regulator of the enzyme. So at that time, we do not require the glycolytic pathway operated. So this how the glucagon regulates the glycolysis and gluconeogenesis. If the glucose level is high, this reaction will be totally in the reversible mode, and instead of a glucagon, the insulin will play a role in the regulation of the level of glucose and rate of glycolysis and gluconeogenesis. Now we'll skip this glycoxylate pathway. We'll discuss into the phase after the completion of a citric acid cycle, because citric acid cycle is in the connection with the glycolysis and gluconeogenesis. So the Krebs cycle, it is tricarboxylic acid cycle, which is the hub of the metabolic system, because it is the central metabolic activity which generate the intermediates responsible for the operation of the different activity of the different pathways of protein metabolism, glucose metabolism, fatty acid metabolism, and nucleic acid metabolism. So it is called the Krebs cycle, where the tricarboxylic acid is the central molecule. It accounts for the majority of carbohydrate, fatty acid, and amino acid oxidation. It also accounts for a majority of the generation of these compounds and others as well. In 1930, it was established that the addition of lactate, acetate succinate, malate, alpha-ketoglutric acid, and citrate and isocitrate, when added to muscle, means that they stimulate oxygen consumption and release the CO2, carbon dioxide. So Albert UG showed that succinate to the fumarate, fumarate to the malate, and malate to the oxalate. And Carl Mauritius and French Noob showed that citrate is converted into the cis-aconitate, cis-aconitate to the isocitrate, isocitrate to the alpha-ketoglutarate, then succinate to the fumarate, fumarate to the malate, and malate to the oxaloacetate. So this is the historical work, and generally we know the carboxylic cycle as the Krebs cycle. Hans Krebs, who discovered this cycle, and he gave the remarkable theory, the principles of the conversion of different molecules by the TCA cycle. But these are the scientists who have given the base of the TCA cycle by the different experiments. So Matthias and Knoop showed that pyruvate and oxaloacetate could form a citrate known enzymatically by the addition of a peroxide under the basic condition. And Krebs showed that succinate is formed from fumarate, malate, or oxaloacetate. This is interesting since it was shown that the other way worked as well. So pyruvate can form citrate enzymatically, that is pyruvate plus oxaloacetate to the citrate, and the interconversion rate of the intermediate was fast enough to support respiration rates. So I think all of you are known about the story of the Rosalind Franklin and this Watson and Crick, who have been awarded Nobel Prize for the discovery of the DNA structure. The actual work was from the other scientist, and finally the credit was given to this Watson and Crick. Same way, the scientist who worked basic in the TCA cycle discovery were the other, and then finally Krebs is given the credit for the discovery of the TCA cycle. So this is the overview of the glycolytic metabolism, glycogen to the glucose, pyruvate, acetyl-CoA, and 2 molecules of the CO2. So overall, if we see in the science, the value of report, once you report first, you will be given the credit. So even though the different discoveries, which was first given with the experimental proof by the different scientists, the credit is not actually given to the scientist for a particular discovery. Same way in the case of the Gregor Mendel and Wallace theory, who have given the earlier aspects before the establishment of this the genetics and the gene-based experiments in the P. So the citric acid cycle enzymes are found in the matrix of mitochondria. The substrates have to flow across the outer and inner parts of the mitochondria. In this picture, this is about the inner mitochondrial membrane. outer mitochondrial membrane and the cytosolic space, inner membrane space. There are infoldings in the mitochondrial membrane, outer surface, inner face, then its particle size, and how these particles are arranged in the membrane, and it is responsible for the reaction. Nathan Kaplan and Fritz Lipman discovered coenzyme A, and Ocha and Lenens that acetyl-CoA was intermediate from pyruvate to citrate. So the fate of acetyl-CoA was given by this 4 different scientists, which is happening into the TCA cycle. So this is the citric acid cycle picture, where pyruvate to acetyl-CoA by the enzyme pyruvate dehydrogenase, then acetyl-CoA to the citrate with the citrate synthase, then citrate to the cis-aconitate by the aconitase enzyme. One H2O molecule will be released, then further the H2O will be added into the reaction, then aconitate to the isocitrate. So aconitate is the intermediate molecule from citrate to the isocitrate molecule. So isocitrate further will be converted into the oxalosuccinate, then the alpha ketoglutarate to the succinyl CoA. So the enzyme involved the isocitrate dehydrogenase to convert isocitrate to the oxalosuccinate to the alpha ketoglutarate, and then succinyl CoA, then finally succinate. Succinate dehydrogenase enzyme convert the FAD into FADH2. Succinyl synthetase enzyme converts GTP into GTP, and the alpha ketoglutarate dehydrogenase enzyme is responsible for the formation of the NADH. So here in this up to the 6th step, 1 and 2 NADH molecule, 1 GTP molecule, and 1 FADH2 molecule is produced. Sorry. Now the succinate is converted into the fumarate, and from fumarate to malate to oxaloacetate. So in this step also, 1 NADH molecule is formed. Now 1 NADH molecule is responsible for the 2.5, or in some of the book it is written as 3 ATP molecule. And this form the FADH2, that is about to form 1.5 ATP molecule. So actually it is 2 ATP molecule is formed. CoA, that is acetyl-CoA, acts as a carrier of acetyl group. So acetyl-CoA is a high energy compound. The delta G0 is for the hydrolysis of thioester is minus 31.5 kilojoule per mole, and it's making it greater than the hydrolysis of ATP. So pyruvate dehydrogenase converts pyruvate to acetyl-CoA and carbon dioxide. Pyruvate dehydrogenase enzyme, that is the multi-enzyme complex, are groups of non-covalently associated enzymes that catalyze the 2 or more sequential steps in a metabolic pathway. So in this comparative table between the E. coli and yeast, the molecular weight of the enzyme is 4.6 kilodalton, and pyruvate dehydrogenase E1 subunit, that is for 24 and 60 dihydrolipoyl transacetylase E2 2460, dihydrolipoyl dehydrogenase, that is E3 12 and 12. So this is about the structural difference of these three subunits, and first one is the E2, then E1 orange, E3 red, and A and B is together in the C. As per this electron XRD and electron microscopy images, EM based image of the core E2 from yeast pyruvate dehydrogenase, 60 subunits associated as 20 cone-shaped trimers that are verticities of the dodecahedron structure. Why such a complex set of enzymes are required in the metabolic pathway? Enzymatic reactions rates are limited by diffusion. With shorter distance between subunits, an enzyme can almost direct the substrate from one subunit to the another subunit. And channeling metabolic intermediates between successive enzymes minimizes side reactions. And third one is the reactions of multi-enzyme complex can be coordinately controlled. So when we imagine the metabolic pathways operated in the cytoplasm and in mitochondria, the enzymes are floating in the cytosol, and the transfer of substrate molecule from one place to the another place is occur by the diffusion only. Now diffusion is a passive reaction without the involvement of the energy. So rate of a diffusion depends upon the concentration of the substrate. If concentration is high, the rate of diffusion is high. So to keep the rate of reaction constant in the metabolic activity, the flow must be maintained. And if the subunits will be present in the cytosol or in the mitochondria at a distant places, flow cannot be regulated. Another point is about its loss in the non-specific reaction. So intermediates which are formed in the one metabolic pathway can be diverted into the other reaction, because the intermediates in the one metabolic pathway could be the intermediate of the other metabolic pathway. That's why it is completely a web of the metabolism. To control the non-specific reaction, it must be channelized from one place to the other place without any physical boundaries. So that's why the multi-enzyme complex is formed. It is required in this reaction step. And third point, that is about the coordination control. So it is like you can say the CNC machine for making the object. So one mold will be formed, and then this mold will transfer to the other machine, then the another subunit will be attached, and then it will go to the third, and then fourth, and finally the product will be formed. So here in the TCA cycle, multi-enzyme complex is like a CNC machine with the different objects. So once the one intermediate will enter in the multi-enzyme complex, it will convert it into the different forms, and finally the stable product will be come out from the enzyme complex. So to keep the coordination control as well as the regulation of the enzyme complex, this system is operated, and it is regulated completely in the cell. Then the covalent modification of the eukaryotic pyruvate dehydrogenase enzyme. Here the pyruvate dehydrogenase phosphatase enzyme convert the inactive form into the active form because the phosphate will be released from the enzyme, and pyruvate dehydrogenase kinase enzyme converts the active to the inactive form. So phosphorylation will inactivate the enzyme and dephosphorylation activates the enzyme. The 5 reactions of the pyruvate dehydrogenase multi-enzyme complex are pyruvate to the this acetyl dihydrolipomide with the intermediate step of hydroxymethyl DPP. So DPP is converted to hydroxymethyl DPP, pyruvate dehydrogenase enzyme. Then lipoamide by the enzyme dihydrolipolyl transacetylase, that is E2. Then the E3 enzyme, that is dihydrolipolyl dehydrogenase enzyme. And finally the NADH is formed. Here in the acetyl CoA will be formed. So pyruvate to the acetyl CoA conversion is controlled by the 3 subunits E1, E2 and E3 of the pyruvate dehydrogenase enzyme complex. That's why this is the most complex reaction in the TCA cycle that happens with the multi-enzyme complex. So the enzyme requires 5 coenzymes and 5 reactions. Pyruvate to the acetyl CoA with the help of pyruvate dehydrogenase. So this is the same reaction which we have discussed in the last slide. This is about the coenzyme, prosthetic groups, cofactor, its location and function of the pyruvate dehydrogenase. Thymine bound to the E1 reaction is decarboxylation, pyrophosphate and pyruvate. Actually, the formatting got changed in this slide. That's why this the name of this cofactors, location and function is got changed in the column. Now we'll discuss the purine nucleotide metabolism and anabolism. Purine nucleotide biosynthesis, that is de novo biosynthesis of the purine nucleotide. So this is the general purine ring structure, 2-ring structure, 4 nitrogen molecules, 2 purine, adenine and guanine, 3 pyrimidine, cytosine, thymine and uracil. Uracil is exclusively present in the RNA and thymine is in the DNA. The rest of the other nucleotides are present in both DNA and RNA. Pyrimidine is the single ring structure, purine is double ring structure. Nucleoside and nucleotide. DNA, RNA, nucleic acid are made up of nucleotide, that is nucleoside is nitrogenous base ribose, nucleotide is nitrogenous base ribose and phosphate. So the phosphorylated nucleoside is the nucleotide, and once the oxygen is removed from the second position, it becomes a deoxyribonucleoside. The structure of nucleotide is, this is for the ribose, not for the deoxyribose. If it is a deoxyribose, the second position where the hydroxyl group is present will be removed, so there will be no oxygen. The reason is, if oxygen will be present in the second position, then it will interfere in the stability of the helical structure and its coiling in the nucleus. So as we know that the nucleic acid DNA is super coiled and packed into the nucleus So if oxygen will be present, the oxygen of second position will form a hindrance with the oxygen of the opposite strand of the DNA, and the structure cannot be stabilized in the nucleus in the supercoiling form. That's why the oxygen is removed from the ribose structure. So there are two pathways leading to the biosynthesis of nucleotides. De novo biosynthesis pathway, this is main synthetic pathway. Biosynthesis of the nucleotides begins very new with the use of small metabolic precursor as a raw material. And in the salvage pathway, already it is on the basis of the precursor molecule or the intermediate molecule. So in the salvage pathway, synthesis of a nucleotides by the recycle of the free nitrogen bases or nucleosides released from the nucleic acid breakdown. This is important in brain and bone marrow. Predominantly, this biosynthesis occurs in the cytosol of liver and to some extent in the small intestine and thymus. In humans, all necessary enzymes of a purine nucleotide biosynthesis are found in the cytoplasm of the cell. De novo biosynthesis occurs in most of the cell's cytoplasm except human brain, polymorphonucleonucleoside, and erythrocytes. Purines are synthesized using 5-phosphoribose as the starting material step by step, and phosphoribosyl 1 pyrophosphate is an active donor of the ribosyl 5-phosphate. The purine ring is synthesized by a series of biochemical reactions that add the carbon and nitrogen atoms to the preformed ribose 5-phosphate. The ribose 5-phosphate is synthesized as part of the hexose monophosphate pathway, that is HMP shunt. So in the HMP shunt, that is source of ribose 5-phosphate, the glucose 6-phosphate to the fructose 6-phosphate to the fructose 1,6-bisphosphate to the dihydroxyacetone phosphate and glycerolate 3-phosphate finally to the pyruvate, that is into the glycolytic pathway. Now in the HMP shunt, the glyceraldehyde 3-phosphate forms the xylulose 5-phosphate, from xylulose 5-phosphate to the ribulose 5-phosphate, from ribulose 5-phosphate to the ribose 5-phosphate. That will go into the nucleotide biosynthesis. Fructose 6-phosphate directly go into the glyceraldehyde 3-phosphate formation, and from that the ribose 5-phosphate. Fructose 6-phosphate go into the erythrose 4-phosphate, pseudoheptulose 7-phosphate, xylulose 5-phosphate, then ribulose 5-phosphate. So because of the shunting of a molecule from one pathway to the other pathway to the third pathway, that's why it is called HMP shunt. The shunting is the phenomena we generally know it from the shunting of a railway's train. So train is the shunting occurs from one railway track to the other railway track to the third railway track because of the traffic onto the different tracks. That's why the trains are diverted. So from this phenomena, the name shunt is given to the this HMP pathway. That is the source of ribose 5-phosphate, and then ribose 5-phosphate will go into the nucleotide biosynthesis, phosphoribosyl pyrophosphate. So this PRPP is the starting material for the protein de novo biosynthesis, and it is formed from the ribose 5-phosphate. So the pentose sugar is always a ribose which may be reduced to deoxyribose after nucleotide synthesis is complete. So PRPP involved in the pyrimidine nucleotides also, and with the help of NAD plus and histidine biosynthesis. So here in the reaction, the ribose 5-phosphate is converted into the 5-ribosyl 1-pyrophosphate. PRPP synthetase enzyme convert the ribose 5-phosphate to the 5-ribosyl pyrophosphate. Activator is the phosphate, that is the phosphorylation ATP. Inhibitors for the purine nucleotide, and ATP is converted into the AMP. And the de novo biosynthesis of a purine nucleotide means a very new synthesis using a raw materials as a phosphoribose amino acid like glycine, glutamine, and aspartic acid, 1 carbon units, and carbon dioxide. Nitrogen and carbon source of purine bioring synthesis. Now this is the slide you have to remember by any means, because in the competitive exams, specifically in NET, many times questions have been asked that which is the source of N1, source of N3 and N9, C2, C8, like that. So the questions based on the source of carbon and nitrogen in the nucleotide ring have been asked in this examination, either in the 4 mark or in the 2 marks, generally into the 2 marks. If the question is very straightforward, like which is the source of N3 and N9, so this kind of question will be of a 2 marks. And if question will be twisted in the form of some experiment, I hope you all have referred the old question papers of the NET examination, so you might have an idea that what kind of the structure of questions will be there in the 4 mark question. So in that the questions will be elaborated in form of experiment, and then the finally you have to give the answer. So this is one of the important slide in terms of the exam questions. Aspartate, CO2, glycine, amide. N1 is from the aspartic acid. Then N3 and N9 from the glutamine, green color, yellow color is aspartate. C2 and C8 is from the tetrahydrofolate. C2 and C9, light blue color. Then C4, C5 and N7 from the glycine. C6, that is from the CO2, that is light violet color. So the donor of the carbon tetrahydrofolate. So in the step 1, formation of the inosine monophosphate, that is IMP, and in the state 2, conversion of IMP to the either AMP or GMP in the de novo stages. IMP, that is inosine monophosphate, is the first biosynthesized purine nucleotide in this de novo synthetic pathway, and IMP is a nucleotide with hypoxanthine as nitrogen base. IMP is then converted to AMP and GMP. Biosynthesis of this IMP molecule. Basic pathway for de novo biosynthesis of purine ribonucleotide starts from the ribose 5-phosphate, and that is 11th step reaction occurs in the liver cytosol. In the first step, that is activation of phosphoribosyl pyrophosphate, that is PRPP. Second and fifth is the entry of a glutamine. Third is the entry of glycine. 4 and 10 is the entry of folate. 6 is the ring closure. 7 is the entry of CO2. 8 is the entry of aspartate. Now, why this sequence is given? Because this is in the examination also a kind of a questions have been asked that at which step which molecule is involved, or what is the source of the third step in the nucleotide biosynthesis. So like if it is third step, then it's the glycine. If it is seventh, entry of CO2, like that. Step number 9, removal of fumarate, and step number 11, that is again ring closure. So in the first step, ribose 5-phosphate plus ATP, PRPP synthetase, PRPP and AMP. PRPP plus glutamine by the amidotransferase, and then the reaction occurs. Once the phosphoribosyl amine is formed, the building block of the purine ring structure begins. In 9 successive reactions, the first purine nucleotide formed is IMP, that is inosine monophosphate. The first step is the activation of ribose 5-phosphate, that is regulatory step or the committed step. That means the reversible step is not occur here. So in the first step, ribose 5-phosphate is converted into the PRPP by the PRPP synthetase enzyme. In the second step, that is acquisition of a purine atom N9, that is by the glutamine. So in the second step, glutamine is converted into the glutamate and the nitrogen will be added. In the third step, that is acquisition of a purine atom C4, C5 and N7. So this atom is donated by the glycine molecule in the third step, and glycinamide nucleotide is formed. In the fourth step, that is acquisition of the purine from C8. C8 is from the folate, tetrahydrofolate, and formyl tetrahydrofolate molecule by the GR transformylase enzyme. Then the acquisition of the third nitrogen in the fifth step by the glutamine. In the sixth step, closing of the imidazole ring, and this imidazole ring is now the base. Seventh step, that is acquisition of the carbon number 6. In the step 8, that is acquisition of the nitrogen 1, first number of nitrogen enters by the donated by the aspartate in the reaction step number 8. It is synthetase enzyme. In the 9th step, that is elimination of the fumarate, and in 10th step, that is acquisition of carbon number 2 by the again N-formyl tetrahydrofolate, that is ICAR transformylase by the enzyme. In the step number 11, that is ring closure to form IMP. So IMP is a nucleotide of nitrogen base, that is hypoxanthine. IMP is the first purine nucleotide synthesized in de novo synthesis mechanism. So this is the overall step of the de novo pathway of a purine biosynthesis. So total 6 ATPs are required in the purine biosynthesis from ribose 5-phosphate to the IMP. Since one step ATP is converted to the AMP, hence this is really 7 ATP equivalents. Now conversion of IMP to the AMP and GMP. Aspartate and GTP is used for the AMP synthesis. Glutamine and ATP is used for the GMP synthesis. These are for the 2 marks question. So that you have to remember in a way that AMP aspartate, in this way you can remember aspartate AA, AMP synthesis, aspartate. Glutamine, GMP, GMP, glutamine, right? G to G, like that. That you have to remember. Then regulation of the purine nucleotide biosynthesis. Purine nucleotide biosynthesis is well regulated to meet the cellular demand. Two enzymes are the key regulatory enzymes for the purine nucleotide de novo biosynthesis. PRPP synthesizing the PRPP, that is feed forward activator. PRPP is for the feed forward activator of PRPP glutamyl aminotransferase. The intracellular concentration of PRPP regulates the purine biosynthesis, and more availability of PRPP increases more synthesis of purine nucleotides if the enzyme PRPP synthesis is not inhibited by feedback control. IMP, AMP and GMP availability to sufficient concentration inhibits the regulatory enzyme by feedback mechanism. So IMP, AMP and GMP is a negative regulator and PRPP is the positive regulator. So that's why it is feed forward, and this is feedback inhibition and feed forward activation. PRPP activates PRPP glutamyl aminotransferase. IMP, AMP and GMP inhibit PRPP synthetase enzyme. Sufficient AMP, that means AMP level is sufficient enough in the cell to drive the nucleotide metabolism, then it will inhibit the conversion of IMP to the AMP. And if GMP is sufficient, it also inhibit the conversion of IMP to the GMP. Reason is, if AMP will be sufficient, that means the synthesis of DNA is not occurring. So that's why AMP level will remain in the cell. That's why it will inhibit, because now AMP is not consumed into the nucleic acid synthesis. That's why this is feedback inhibition by the AMP and GMP. Regulation of AMP synthesis, that is adenylosuccinate synthetase, is feedback inhibited by the AMP. Regulation of GMP synthesis, that is IMP dehydrogenase enzyme, is feedback inhibited by the GMP. ATP stimulates conversion of IMP to the GMP. GTP stimulates conversion of IMP to the AMP. Here, in terms of ATP and GTP, you have to remember in crisscross manner, like ATP to the GTP, that is GMP, and GTP to the AMP. In terms of amino acid, it is A to A, G to G. In terms of energy, it is cross A to G and G to A. This way you can remember the regulatory molecules with respect to the questions, and that ensures the balanced synthesis of both families of purine nucleotide. Significance of regulation of de novo synthesis, that meet the sufficient need to the nucleotide to body function without the wasting. AMP and GMP control their respective synthesis from IMP by feedback mechanism GTP to ATP. So this is regulated by the feedback inhibition, that is ribose 5-phosphate to the alpha phosphoribosyl pyrophosphate, then phosphoribosyl beta 1 amine, and to IMP to the AMP and GMP. So as per this figure, the GMP, GDP and GTP, all these three molecules inhibits the activity of ribosyl 5-phosphate pyrophosphokinase. That is the first step, and then it also inhibits the aminotransferase enzyme. Same way, the AMP, ADP and ATP inhibits these two enzymes. These three molecules also inhibit the adenylosuccinate synthetase. It also inhibits the IMP dehydrogenase enzyme. So in the feedback inhibition mechanism, the AMP, ADP, ATP as well as GMP, GDP and GTP inhibits these 3 and 4 enzymes. 2 are common: ribose 5-phosphate pyrophosphokinase and deamidotransferase. So here we keep this lecture up to this because it's almost 12 o'clock. So the rest of the topics I will cover it in the next lecture. So I keep up to this purine biosynthesis and its regulation, because since the metabolism is very large subject, it is very difficult to complete it within the 2 hours and 4 hours of time. That's why I have divided into this 2 portion. I hope I have tried to justify the topic within the short time, and whenever just through my past experiences and through the examination, I had some idea about the metabolic pathways. Generally, the questions which have been asked in the net examination and other competitive examinations. So I have tried to discuss those questions in the lecture also. I hope I have tried to justify the subject. So I keep up to this, and thank you for listening this lecture.